Welcome to Psychology 311, Study Session 13, Chemical Senses. 1. Olfaction. Introduction. Chemical perception of taste and smell. When we eat or drink, we perceive a sensation that most people call taste. However, we all know that when the nose is blocked, for instance, when one has a common cold, the sensation is considerably reduced. Welcome to Psychology 311, Study Session 13, Chemical Senses. 1. Olfaction. Introduction. Chemical perception of taste and smell. When we eat or drink, we perceive a sensation that most people call taste. However, we all know that when the nose is blocked, for instance, when one has a common cold, the sensation is considerably reduced. This is because it results from a combination of stimulation of chemical receptors, chemoreceptors, in the nose as well as the mouth. The chemoreceptors in the mouth are called gustatory receptors, and those in the nose are olfactory receptors. The sensations that result from individual stimulation of these two types of chemoreceptors are respectively taste and smell. Chemoreceptors are not, however, the only sensory receptors involved in the appreciation and discrimination of food and drink. At least two other modalities of sensation affect the overall experience. The smoothness, texture, and crunchiness of food are conveyed by mechanoreceptors on the tongue and throughout the rest of the mouth, including the teeth and in the pharynx. Thermoreceptors in the mouth also detect the temperature of solids and liquids. Just think of the combination of experiences: touch, fizziness, coolness, acidity, and exquisite smell that make up the experience of drinking champagne. Those who like spices in their food even derive pleasure from the stimulation of receptors normally involved in the sensation of pain, nociception. Chemicals found in common spices, such as chili peppers, stimulate these nociceptors in the mouth, and the resulting sensation is referred to as the common chemical sense. The taste or smell system fulfills two separate physiological roles. Not only does it help us to identify good food containing essential nutrients, salts, carbohydrates, proteins, and fats, but it also provides a warning of the unsuitability of harmful and potentially toxic substances by detecting them before they are ingested. Objectives. At the end of this lecture, you should be able to 1. Describe the chemical senses. 2. Explain the perception of taste and smell. And 3. Explain the mechanism of smell and olfaction. Pre-test. 1. What is olfaction? 2. Identify the chemical senses. 3. Describe the functioning of the chemical senses. Olfaction. The nose is the organ responsible for the sense of smell. The cavity of the nose is lined with mucous membranes that have smell receptors connected to the olfactory nerve. The smells themselves consist of vapors of various substances. The smell receptors interact with the molecules of these vapors and transmit their sensations to the brain. The nose also has a structure called the vomerosenal organ, whose function has not been determined, but which is suspected of being sensitive to pheromones that influence the reproductive cycle. The smell receptors are sensitive to 7 types of sensations that can be characterized as camphor, musk, flower, mint, heather, acrid, or putrid. The sense of smell is sometimes temporarily lost when a person has a cold. Dogs have the sense of smell that is many times more sensitive than man's. Mechanism of smell and the olfactory receptors. The human olfactory organ, the olfactory epithelium or mucosa, is a sheet of cells 100 to 200 millimeter thick, situated high in the back of the nose cavity and on the thin bony partition, the central septum of the nasal passage. The olfactory system responds to airborne, volatile molecules that gain access to the olfactory epithelium with the in and out airflow through and behind the nose. The odor molecules are distributed over the receptor sheet in an irregular pattern by the turbulence of the airflow set up by the turbinate bones in the side walls of the nose. The molecules diffuse through the surface layer of mucus and stimulate the olfactory receptors. Hydrophilic, that is water-soluble molecules, dissolve readily in the mucus, but the diffusion of less soluble molecules is assisted by odor-binding proteins in the mucus. These odor-binding proteins are also thought to assist in removing odor molecules from the receptor cells. The mucous layer moves across the surface of the olfactory mucosa at 10 to 60 millimeters per minute toward the nasopharynx, the continuance of the nasal cavity backwards and downwards to link to the pharynx. This flow of mucus, which is increased and becomes more watery in such conditions as infection of the nasal cavity and hay fever, also assists in the removal of odors after they have been sensed. The olfactory epithelium contains specialized elongated nerve cells, olfactory receptors. These cells have very thin fibers that run upwards in bundles through perforations in the skull, the cribriform plate, above the roof of the nasal cavity, below the frontal lobes of the brain. These bundles of nerves constitute the first cranial nerve, the olfactory nerve. They extend only a very short distance, ending in the olfactory bulbs, which are a pair of swellings underneath the frontal lobes. The other end of each olfactory receptor, pointing down into the nasal cavity, is extended into a long process, ending in a knob carrying several hairs, cilia. These cilia are bathed in a thin layer of mucus secreted by specialized cells in the olfactory epithelium, in which the molecules of odorous substances dissolve. In the membrane of the cilia are olfactory receptor proteins, which interact with the smelly molecules and initiate a cascade reaction inside the cell that leads to a change in the rate of impulses, action potentials, passing along the nerve fiber. Human beings are able to distinguish 10,000 or more different odors. There have been valiant attempts to classify these into a smaller number, usually 10 to 20, basic or primary smells comparable to the 4 or so primary tastes, but no scheme is universally accepted. The human nose, not to mention that of a sniffer dog, can be incredibly sensitive to very low concentrations of odorous substances. Certain male moths use similar receptor cells on their antennae to detect even single molecules of a pheromone secreted by female moths. Individual olfactory receptor neurons fire off spontaneously at between 3 and 60 impulses per second. When stimulated with particular odors, they increase their firing frequency. Each receptor cell responds, but not equally, to many different types of odor. As in the gustatory system, the successive nerve cells in the pathway become more selective, each responding to fewer odors. Interestingly, despite the poor selectivity of individual receptor cells, different regions of the olfactory sheath, consisting of hundreds or thousands of receptor cells, are maximally responsive to particular odors. The overall pattern of activity in the olfactory epithelium can be mapped with electrical recording methods, electro-olfactogram, or other techniques for detecting active regions. Each distinctive odor produces its own fingerprints of activity across the epithelium. This mapping is thought to reflect the pattern of expression, that is, activation of genes that make the receptor proteins in the receptor cells membranes. A huge family of odor receptor genes exists in the mouse, perhaps as many as 5% of all the genes. The spatial coding of odor equality is transmitted to the first relay of the olfactory pathway. A huge family of odor receptor genes exists in the mouth, perhaps as many as 5% of all the genes. The spatial coding of odor quality is transmitted to the first relay of the olfactory pathway, the olfactory bulb. There is a loose topographical projection from the receptor sheath to the bulb, where the axons form synapses with neurons called mitral cells. The olfactory bulb contains a complex network of nerve cells and is responsible for a considerable amount of sensory processing. Hence, neurons in the olfactory bulb respond with one distinct temporal pattern of impulses to one odor and different patterns to another smells. The mitral cells send their fibers into the olfactory tracts, which runs backwards. Some ends in the thalamus, which in turn send fibers up to the olfactory cortex. The neurons of the olfactory cortex are still not highly specific for particular odors. Other fibers of the olfactory tract have direct connections to areas of the limbic system around the region of hypothalamus. Since the limbic system is thought to be responsible for regulating emotions, this might explain the fact that smells can evoke strong feelings of enjoyment or aversion, the hedonic component of sensation. Unlike other stimuli, olfactory stimuli are not very time dependent. The effect of visual, tactile, and auditory stimulation follow the stimulus immediately, whereas some olfactory stimuli, such as those left by animals when marking their territory, remain when the animal is long gone. In this way, olfactory stimuli and their behavioral and social effects can have more lasting consequences. The olfactory system occupies a smaller fraction of the brain in humans than in many other species, and this is part of the evidence for the commonly held belief that people are generally inferior in their sense of smell. Studies in other animals, from insects to hamsters to monkeys, have revealed the importance of olfaction for many aspects of behavior, especially reproduction. For example, male rhesus monkeys use smell to sense the hormonal status of females, ovulating or not, with a marked effect on their level of sexual activity. But when in humans, there is growing evidence that olfaction, mainly unconscious, is important in such functions as sexual preference and recognition of other people. Summary. In this lecture, we have learned of the olfactory system. The structure revealed that olfactory system is one of the most vital parts of the brain responsible for the sense of smell. In some animals, it is the primary mode of communication and influences many important functions, including reproduction and taste. The nose contains specialized sensory nerve cells or neurons which sends a nerve fiber called an axon to the olfactory bulb, a brain structure just above the nose. We also learned about the mechanism of smell and the olfactory receptors, as well as how the spatial coding of odor quality is transmitted through the olfactory pathway. This is the end of study session 13. Thanks for listening.